Why, hello! We are going to dive into chapter 12, which is the heart and cardiovascular system. So let's go ahead and get started. Looking at, here we go, chapter 12. Flip through these learning outcomes. They're already posted for you in your chapter guide and in the weekly folder. The sun is like gonna shine. See, it's like bright shining into my window right there. Sorry about that. Anyways, all right. Let's talk about the heart, where it's located and what makes it up. It's basically, in the mediastinum, slightly left of the center line of the body. It's rotated left as well. The base is the superior part of the heart, so it's kind of like an upside down triangle. The apex is the inferior tip. It's pointing down towards the left, and it has great arteries and great veins coming off of it, as we mentioned a little bit in the last lecture. There it is in its relative location. Has a superior border, a right border, a left border, an inferior border. And here you can see some of those borders, right, left, inferior, superior. Let's talk about the layers of the heart. The epicardium is also the visceral pericardium. The visceral pericardium is part of the serous membrane that surrounds the heart. You have the visceral and parietal pericardium. So the visceral is on the surface of the heart. It's called the epicardium. It's attached to the myocardium itself. The myocardium is the muscular wall of the heart. It contains the blood vessels and nerves that go into the heart, not into the chambers of the heart, but that are feeding the heart muscle itself. Then you have the endocardium. The endocardium is a simple squamous epithelium that's lining the very inside chambers of the heart, including the valves. Let's talk about that pericardium. I already mentioned the visceral pericardium, but there's also a parietal pericardium that's reinforced by a very fibrous layer. If I were to cut your chest open now, I would basically see your heart in a big, fatty, fibrous case. The fat surrounds your heart for insulation and protection, and then the fibrous case would be that visceral or that parietal pericardium. The pericardial cavity is between the visceral and parietal pericardium, and it's filled with a serous fluid, pericardial fluid, basically just there to reduce friction between the two layers. So you have your parietal pericardium, which is very thick, fibrous. Then you have this pericardial cavity with the serous fluid, the pericardial fluid, the epicardium, myocardium, endocardium is the deepest layer. Let's talk about the atriomyocardium. It's relatively thin. The bundles, it's basically little muscle bundles that wrap around the atria, and then the ventricular myocardium is more spiral shaped instead of just circular shape. It kind of tips around and wraps around all the way down towards that apex, towards that tip. So as you can see, the atria are individually wrapped, and then the ventricles are kind of wrapped and spiraled all together. Let's talk about the cardiac muscle tissue. Cardiac muscle tissue is the cells themselves. The cardiocytes are smaller than regular skeletal muscles. They have essentially located nucleus instead of being multinucleate like skeletal muscles. They also are branched as opposed to skeletal muscles, which are just thin fibers. They have special intracellular connections called intercalated discs. There you see them. You can see this is like one big cell here, and then it branches here, and then this one kind of branches this way and that way. They kind of branch and reform together, and the intercalated discs are the special connections between them. Cardiac muscle cells are found only in the heart. They're organized into, or they contain myofibrils, just like skeletal muscles, and they have sarcomeres, just like skeletal muscles, so it's the same basic setup. They're dependent on aerobic metabolism, so they have to have oxygen in order to contract. They contain lots of mitochondria and very high levels of myoglobin. Myoglobin is the pigment that stores oxygen. So if you ever are deprived of oxygen for whatever reason, the myoglobin will be there to sustain your heart muscle for a little bit. There are the cardiac cells, the myofibrils, intercalated discs, etc. Let's talk about those intercalated discs. They're basically specialized gap junctions. They allow ions and small molecules to move from cardiocyte to cardiocyte. They're also directing electrical connection between the cells. I imagine the cardiocytes as like little cells that hold hands, and they say, okay, 1, 2, 3, contract. 1, 2, 3, contract. So they're all kind of holding hands and contracting together. It helps to coordinate the movements. There's also desmosomes, which are special connections to help anchor the cells together. Here you see intercalated discs and those special desmosomes linking them together. You can pause the video here, go back and review this little section. The heart is located within the mediastinum. That's the area between the two pleural lung cavities, and it's on the anterior portion. It's surrounded by the pericardial sac, as we've talked about already. There's lots of blood vessels in this area, including the superior inferior vena cava, the aorta, the thymus, the esophagus, the trachea. Lots of organs in there. So again, it's just this kind of middle area. Let's talk about the pericardial cavity. The pericardial cavity is kind of like a fist being pushed into an inflated balloon. The fist is like your heart. The wrist is like the base of your heart where all the great vessels are. And then the air space between the balloon is that pericardial cavity. The pericardial cavity has pericardial fluid. Pathogenic infection can cause pericarditis, which is the inflammation of those pericardial surfaces. And it produces like a scratching sound that you would hear through a stethoscope. So here's what we mean. The heart is kind of like in a double bag. Let's talk about the exterior features of the heart. On the anterior surface, you see the auricle. The auricle is off of the right and left atria. It's just an extra expandable area of the atria. The sulci are the shallow grooves or the boundaries in between the chambers. There's a coronary sulcus and an anterior interventricular sulcus. This is not only a boundary line, but this is also where the blood vessels are going to sit and kind of be nestled into our heart structure. The ligamentum arteriosum is the little attachment. It's a ligament between the pulmonary trunk and the aorta. It's a remnant of the fetal connection between the pulmonary and aortic vessels. Here you see ligamentum arteriosum. Here you can see some of the sulci. And again, the sulci are where the blood vessels are. So they're normally just grooves, but now they're grooves that are filled with blood vessels. I didn't point out the auricle, just extra expandable tissue. On the posterior surface of the heart, you have the left atrium connecting to the pulmonary veins. You have the right atrium connecting to the coronary veins. You have the coronary sinus, and you have the posterior interventricular sulcus, which is where the posterior interventricular vessels are. You can pause the video here, go back and answer these 3 questions. Let's talk about how the heart gets blood supply. So this is called the coronary circulation. The coronary circulation is referring to the heart's blood vessels. Every organ of your body has blood vessels going to it, so your heart needs it too. The heart is the pump that's going to pump the blood through all of the body organs and body tissues, but it also has to pump blood to itself. Otherwise, the heart tissue would die. So during exertion, blood flow can increase up to 9 times. To that myocardium, because that myocardium is increasing its contractions. Kind of crazy. It almost makes my head hurt to think that my heart is beating every second. I don't know. Maybe it's because I'm not that smart. I don't know. Either way, the coronary circulation is what we call our heart circulation. It begins with the coronary arteries. It begins at the base of the ascending aorta through something called the aortic sinus, and it basically peaks when the heart is relaxed, and it almost ceases when the heart muscle contracts. So you'll see how that works in a second. So that left coronary artery supplies the left ventricle, the left atrium, the interventricular septum. There's something called a circumflex artery that goes around to the back side of the heart, and there's an anterior interventricular artery which goes down that anterior interventricular sulcus. The right coronary artery branches off and forms the marginal arteries and also forms the posterior interventricular artery. And then there's an arterial anastomosis between anterior and posterior interventricular arteries. This is when the arteries feed into each other. And that's, again, another protective measure to make sure that the heart's going to get blood flow, even if one of those vessels is blocked. It's a pretty good thing, right? So our left coronary artery is actually pretty small before it branches. Then it branches and it goes into the anterior interventricular. It's also called the left anterior descending, or LAD. So if you've ever known someone to have a heart attack, it's probably because this LAD, or anterior interventricular artery, has been blocked. The circumflex going around the back, and our marginal arteries coming off of the right coronary artery on the right side, and then going down the back side, turning into our posterior interventricular artery. Someone who has a double bypass surgery is probably having a bypass put in here in the anterior interventricular and the circumflex. Usually triple and quadruple bypasses are involving the circumflex, anterior interventricular, marginal arteries, part of the right coronary artery, and the posterior interventricular. So there would be blockages all over the place. Let's talk about the anterior surface. You have the anterior cardiac veins. They all drain into the great cardiac vein. The great cardiac vein is on the posterior side, and it enters into what's called the coronary sinus on the back of the right atrium. You see all the veins here, ultimately collecting and draining back into the sinuses. The coronary sinus is going to open into the right atrium, and that is how the coronary circulation gets back into the heart to be pumped back over to the lungs to get oxygen. There it is. It's just like a swelling or an enlarged area of that great cardiac vein. So our aorta, our big blood vessel, has elastic rebound associated with it. When the left ventricle contracts, it's going to force blood into the aorta, and the aortic has an elastic wall, so it's going to stretch. When the left ventricle relaxes, the pressure in the aorta declines, and the walls of the aorta recoil, and that's the idea of elastic rebound. It can kind of stretch and contract as it needs to with that increasing blood pressure, decreasing blood pressure. I should stop here for a second. As that recoil is happening, as the blood pressure is lowered, that's when the coronary arteries are going to be supplied with blood. So the coronary arteries don't get that high pressure blood that the aorta has. The aorta takes the brunt of that blood pressure because it's big and elastic. The coronary arteries are relatively delicate, so they're just getting a little bit of backflow from the aorta. You can pause the video here, go back and answer these 3 questions. Let's talk about the chambers of the heart. The blood flows from the atria into the ventricles. The interatrial septum divides the 2 atria, interventricular septum divides the 2 ventricles, and the atrioventricular valves, or AV valves, are the little valves in between the atria and ventricles. There's one on the right and one on the left. These allow for one-way blood flow through the heart. Nothing goes backwards through the heart. Nothing goes from the ventricles to the atria, or at least it shouldn't. If it does, you have a problem. Right atria is receiving that deoxygenated or low oxygen blood from the rest of the body. The fossa ovalis is the location for the foramen ovale in the embryo. The foramen ovale closes at birth in some cases. Sometimes people are born with a hole in their heart, and that's probably what it is. It will eventually close, or surgery can be done to fix it. The pectinate muscles form the ridges of the anterior arterial wall, atrial wall, and the inner surface of that auricle. So they're just there muscles that can help expand and contract that atria as needed. The left atrium receives the oxygenated blood from the lungs. Our right ventricle receives blood from the right atrium through the right AV valve, which is also called the tricuspid valve. The free edge of the valve has 3 cusps or flaps, which is why it's called the tricuspid, and they're connected to these little pieces of connective tissue fibers called chordae tendineae. The chordae tendineae originate in little muscles called papillary muscles, which help control the opening and closing of that AV valve. When there's contraction going on, the blood exits through the pulmonary valve. The pulmonary valve, or pulmonary semilunar valve, takes the blood into the pulmonary trunk. There's something called a moderator band. The moderator band tenses the papillary muscles just before a contraction to prevent those AV valves from slamming shut. The left ventricle has much thicker walls than the right ventricle because it has to produce more pressure. Because the right ventricle is only pumping the blood to the lungs, the left ventricle is pumping the blood to the entire body. So it has to have a little bit more oomph behind it. It receives blood from the left atrium through what we call the left AV valve, or bicuspid valve, or mitral valve. The bicuspid just means it has 2 cusps, unlike the tricuspid. So with the contraction, the blood exits through the aortic valve, or the aortic semilunar valve, into the ascending aorta. The trabeculae carnae are just the muscular ridges on the inner surfaces of the right and left ventricles. There is a video here. It's not attached to this PowerPoint, but you can see it if you visit the textbook website. Here is all of those structures I just mentioned. What I suggest doing, not only following the blood flow through the heart, but also making sure that you note all of these structures. Again, on a test or quiz, I'm not going to give you a diagram, but your lab instructor might, so it's important to know these structures. When the valves, what happens to the valves when the ventricles are relaxed? Basically, the chordae tendineae are loose, the AV valves are open, the blood flows passively from the atria to the ventricles. It's just draining right on down because of gravity. There's pressure in the pulmonary and systemic circuits to keep those aortic and pulmonary valves closed, and blood flows into the coronary arteries, again, when the ventricles are relaxed. They originate in the aortic sinuses, as I mentioned, which is just next to the aortic valve, where that backflow would occur. So let's see, here's our valves when they're open. This is just at rest. Our ventricles are not contracting. Our AV valves are open, and that blood's flowing down freely into the ventricles. Now the ventricles are going to contract. In order for the ventricles to contract, those AV valves have to close. So blood moving toward the atria basically pushes the AV valves together, closing those AV valves. Papillary muscles contract, making sure the AV valves don't burst the other direction and open upwards. They keep them closed. And there's an increasing ventricular pressure. It's kind of like it starts to squeeze, it starts to squeeze, it starts to squeeze. And then once that pressure reaches a certain limit, the contraction and the actual movement occurs. So there has to be a buildup of sustained contraction. The aortic and pulmonary valves, also called the semilunar valves, will respond to that increasing pressure. Blood will flow from the ventricles into the pulmonary trunk and the aorta. Again, this video here is not linked to the PowerPoint, but you can view it by visiting the textbook website. Here you see the whole thing happen. Our AV valves will close. Pressure will build as we have a little bit of isometric contraction in our muscle. This little papillary muscle is going to contract, holding those valves down so that the blood doesn't burst up through the atrium. And then once pressure reaches the threshold, the semilunar valves will open. You can pause the video here, go back and answer these 3 questions. So let's talk about compromised heart function. Coronary artery disease would be when the coronary circulation is completely or partially blocked. It results in reduced cardiac function. So reduced circulatory supply to the myocardium is called coronary ischemia. And atherosclerotic, it's hard to say, plaque is usually the cause, some type of blockage, a plaque, cholesterol buildup, etc. The plaque or the thrombus can narrow the vessel passageway, and then the vessel will start to spasm or kind of freak out and say, oh God, there's not enough blood coming through here, and so it constricts, and that's going to block blood flow, and those pathways or the blockage can be viewed by doing a, oh gosh, what's it called? Catheterization. They do a cardiac catheterization where they just inject a dye into your blood and they trace it through, and if they see part of your coronary arteries are blocked, well, then you have a blockage. And it usually has to be removed through some type of surgery. So here you see this whole area is being blocked. This would be like a normal heart. So other treatments are balloon angioplasty, which is when a little catheter is inserted and the balloon kind of pushes the plaques up against the walls. A stent can be used to prevent the redevelopment of those plaques, just a little wire mesh tube inserted into the vessel to push the wall open to make sure that it doesn't collapse. You can get several stents along the length of a blocked vessel. There is a stent right here. Valves can be compromised as well. Proper function of the valves is necessary for the adequate flow through the heart and to the body. So a valvular heart disease, or VHD, is diagnosed when the heart is unable to maintain a normal blood flow or adequate blood flow. It could be because of congenital malformations or carditis, inflammation of the heart. Can be a treatment as well. The treatment may involve valve replacement. This is the normal valve when it's open, when it's closed, and a diseased heart, damaged aortic valve. See how there's plaques and growths associated with it, preventing it from opening as it needs to. So they can put in a prosthetic valve. Prosthetic valves are typically made from, or they can be taken from a cadaver. They can be taken from a pig or a similar organism, put into your own body. They are doing research to see if they can grow them from stem cells. I know they can grow them from stem cells, but I'm not sure if they have actually put them in a human at this point, the ones that have been grown from stem cells. Either way, heart sounds that are produced. This is what you listen to when you listen through a stethoscope. There's a lub, which is the first sound, and a dub, which is the second sound. The first sound is the closing of the AV valves. The second sound is the dub, and that is the closing of the semilunar valves. So I think of the heart kind of like this, right? You have your AV valves, which are just, atrioventricle, and then you have your semilunar and pulmonary valves, which are kind of at a diagonal going to the lungs and to the body. So AV valves, semilunar valves. And I think of them like this: lub, dub, lub, dub, lub, dub. First, second, S1, S2. And that's just how I remember it, just by doing that little motion. The third and fourth sounds are just from the swishing that occurs in the ventricles and atrials as they contract. It's very faint, usually not heard in a healthy adult. There they are, lub, dub, You can pause the video here, go back and answer these 3 questions. All right, let's talk about the cardiac cycle. 2 main phases. You have contraction and relaxation. Contraction is called systole. This is when the blood's pushed into the adjacent chambers of the arterial trunk. And relaxation is diastole. This is as the chambers are passively filling. So, contraction, relaxation. So, our heart is going to contract differently. The entire heart does not contract at the same time. That would be a huge mess. The atria contract first. So, our heart's relaxed, then our atria contract. This is kind of like wringing a sponge, squeezing the rest down into the ventricles. Then, our ventricles contract. Notice as our ventricles contract, our AV valves are shut. Notice as our atria contract, our semilunar valves are shut. And then, relaxation again. That's the cardiac cycle. It's that easy, kind of. So the atria contract first, pushing blood into the ventricles. It's called atriosystole. While the atria are in systole, the ventricles are in diastole. Both ventricles contract, and then the ventricles are in systole, and the atria are in diastole. Remember, diastole is resting, systole is contracting. So here you see the whole thing diagrammed out a little bit more detail. So this inner line is representing the atria. This outer line is representing the ventricles, and this is just representing time in 800 milliseconds. So it's not even a second that this happens. So we'll start here. Atria contract. As the atria contract, our ventricles are resting. Our ventricles are now done contracting and they start resting. And so our ventricles start contracting. Right at this point, this is when those AV valves shut. As soon as the atria are done contracting, AV valves shut, pressure builds in the ventricles, and then finally the ventricles contract. Then the ventricles relax. Notice the ventricles and the atria are relaxing at the same time for about half of the entire cardiac cycle. So about half of the cardiac cycle, the cell is just at rest, or the heart is just at rest. So cardiac cycle begins when all the chambers are relaxed. Then we have atriosystole. This is when the ventricles are filling. Then we have atriodiastole, which is going to be which is going to continue until the next cardiac cycle begins. Ventricular systole, the first phase is when those AV valves are shut closed, semilunar valves are still closed, and we have this isovolumetric contraction. The pressure's building, but nothing has changed yet. The second phase of ventricular systole is with increasing pressure. We finally have ventricular ejection, and the blood's going to leave through those semilunar valves. Then we have ventricular diastole, where the pressure drops in the ventricles, blood's going to flow backwards, causing those semilunar valves to close. And then isovolumetric relaxation, everything goes back to normal, AV valves are still closed, and the blood flows into the atria. Then towards the later end of ventricular diastole, all the chambers relax, the AV valves finally open, and the ventricles can passively fill until they get to about 70% of their complete maximum volume, and then the atria are going to contract, and the whole thing starts over again. So, cardiac cycle begins. Atriosystole, contraction. Then our AV valves shut. Our isovolumetric contraction begins in the ventricles. All valves are shut at this point, right? Then we have our ejection phase, late ventricular systole. Our AV valves are shut, our semilunar valves are open, our ventricles are contracting, blood's being injected either to the pulmonary and or systemic circuits. Then we have a time of early ventricular diastole. Notice both chambers are resting at this point. Our AV valves are closed, our atria are passively filling. Then our AV valves open, our ventricles fill to about 70% of their resting volume, and then finally the atria can contract, and so on and so forth. The cycle happens over and over again. Our heart also has a conducting system. There's some nerve impulses that have to drive this process. So we have a network of specialized cardiac cells. One's called the SA node or sinoatrial node. This is on the posterior wall of the right atrium. This is our pacemaker. It's the pacemaker that generates the electrical impulses that are needed to conduct the heartbeat. There are internodal pathways which carry the signals through the atrial muscle cells, and then the atrioventricular node, which is the junction between the atria and ventricles. There's also the AV bundle, which is within the interventricular septum. It's only an electrical connection between the atria and ventricles. It contains bundle branches which extend towards the apex, and Purkinje fibers which propagate the action potentials through the ventricular myocardial cells, triggering them to contract during systole. So here's the whole conducting system. SA node, Internodal pathways taking that charge to the AV node traveling down the AV bundle to the bundle branches to the Purkinje fibers where ventricular systole can be initiated. So it is all initiated from the SA node, from that sinoatrial node. So the cardiac muscle has something called automaticity. It contracts on its own without neural or hormonal input. The AV node contains pacemaker cells. If the SA node or internodal pathways are damaged, the pacemaker is still going to generate a pace between 40 and 60 beats per minute. Maximum heart rate is around 230 beats per minute, and this is only reached if there's some type of damage to the conducting system or some type of drug stimulation. You can pause the video here, go back and answer these 3 questions. Let's talk about the nerve control of our cardiac muscle. So it's all controlled through the autonomic nervous system. Obviously, we're not in conscious control of our cardiac muscle. It's basically going to adjust the heart rate and the force of contraction. So we have a cardioinhibitory pathway or center, which is dominated by our parasympathetic neurons, and a cardiac acceleratory center, which is dominated by our sympathetic neurons. So here are our 2 pathways, both located in the medulla oblongata. That's our cardioinhibitory center. The cardioacceleratory center is in our medulla oblongata as well. They're just through different pathways. And again, the parasympathetic is going to slow down our heart, keep our heart nice and slow. Sympathetic is going to increase. Resting heart rate varies with age and physical conditioning, typically between 60 and 100 beats per minute. Condition called bradycardia would be when you have a heart rate that's lower than your normal range. It might be normal for someone else, but low for that person. Tachycardia is faster than normal range. Here you see the general range of resting, tachy, and bradycardia. An EEG or an ECG or an EKG, electrocardiogram, is the recording of the electrical activities of the heart using electrodes that are placed on the body's surface. It can be used to assess conduction, contractile functions, and it can also reveal damage from heart attacks. On the ECG or EKG, you have a P wave, which shows the depolarization of the atria. Before I go on, let me just say that the ECG or EKG is not measuring and it's not telling you when the contraction occurs. It's telling you when the charge changes. It's showing you the depolarization and repolarization. It's not showing you contractions, so don't mix it up. The P wave shows the depolarization. QRS complex is showing you the depolarization of the ventricles, which is why it's a very big change, as well as the repolarization of the atria. The T wave is showing you ventricular repolarization, and those are your waves on the EKG. You also have something called a PR interval, which can dictate or tell you, indicate damage to the AV node, or the QT interval, which would is really used to indicate time between depolarization and repolarization. So here you see what I'm talking about. P wave, this is atrial depolarization. QRS, this is ventricular depolarization, atrial repolarization. T wave is ventricular repolarization. You can pause the video here, go back and answer these 3 questions. So let's talk about cardiac output. This is probably one of the most complex topics of the entire chapter. You're probably like, whoa, really? I thought it was already hard. Yeah, this part, I don't know, it can be straightforward, but you also just have to think logically about it. It's very logical. So the amount of blood pumped by the left ventricle in 1 minute is called the cardiac output. The amount of blood pumped by the left ventricle in 1 minute is called the cardiac output. There are several factors that depends on. It depends on your heart rate, how quickly is your heart beating in that minute, and the stroke volume. How much blood is pumped with one contraction, with one single heartbeat. So heart rate and stroke volume equal cardiac output. Cardiac output is calculated by multiplying heart rate and stroke volume. If you're ejecting 10 milliliters of blood and your heart beats 10 times per minute, you just do 10 times 10, and that's your cardiac output. I hope that's not your cardiac output. Your body can adjust the cardiac output to meet your needs. Your heart rate can increase by up to 250%. Stroke volume can even double. So your body can adjust depending on your needs. Heart failure is when the heart cannot meet the demands of the peripheral tissues. It can be very dangerous. So on average, 75 beats per minute 80 milliliters of blood per beat or per contraction, you're going to get about 6,000 milliliters per minute of blood pumped out of your left ventricle. Kind of crazy, right? 6,000 milliliters, 6 liters, right? That's 3 2-liter bottles every minute. Crazy. All right. So variations in cardiac output. Again, during normal range of exercise, it's going to increase a little bit. Maximum trained athletes can hit peak levels. Other factors affecting heart rate: neuroregulation, sympathetic and parasympathetic stimulation, your body temperature. Increased body temperature and decreased body temperature are going to have an effect on your heart rate. Hormonal regulation, epinephrine, thyroid hormone, all going to have an effect on your heart rate. This is just showing factors that affect heart rate. Then you have factors that affect stroke volume. Venous return is the amount of blood that's returned back to the atria with each heartbeat cycle in the minute. So it's influenced by blood volume, muscular activity, the rate of blood flow through the peripheral capillaries. The filling time is the duration of the ventricular diastole. So in other words, how long that ventricle is sitting there at rest. The longer the filling time means the more blood's going into the ventricles. More blood in the ventricles means a higher stroke volume. This is called Starling's law of the heart. The more time it has to fill, the more it's going to be filled. Just basic logic. So increased venous return equals increased stroke volume. Decreased venous return equals a decreased stroke volume. So again, this is what I said. It's logical, but it's still a lot. There's also contractility. Contractility is the amount of force that's produced by the ventricular contraction. This is regulated through hormones and neural communication. Neural regulation through sympathetic and parasympathetic, and hormonal would be like epinephrine, norepinephrine. Hormonal regulation is typically increasing contractility. Neuroregulation through sympathetic is increasing contractility. Parasympathetic has minimal effect on contractility. Medications can also reduce contractility, especially if you have maybe heart, high blood pressure issues or something like that. You might be taking something to reduce the contractility and stroke volume. So factors affecting stroke volume: contractility, which is affected by neural hormonal regulation, and venous return, all either increasing or decreasing stroke volume. So overall, you have heart rate and stroke volume, which can be influenced by all these factors affecting your cardiac output. You could pause the video here, go back and answer these 3 questions. Let's talk about the influences of cardiac output. So the pulsing of our heart is generating blood pressure. Blood pressure is maintained throughout the whole cardiovascular system. You have arterial pressure, which is much higher than venous pressure. The arterial pressure has to overcome peripheral resistance. You have to get the blood up and out of the heart and to the body in a very effective way. So resistance is going to increase as those vessels get smaller. Capillary pressure is relatively low, which allows a very slow flow of blood in time for capillary exchange. If the blood was racing through the capillaries, capillary exchange couldn't take place. You wouldn't be able to swap oxygen for carbon dioxide. You also have neural and hormonal regulation that's adjusting your heart rate, stroke volume, peripheral resistance, and venous pressure. Venous pressure is going to influence venous return, which is the amount of blood that's arriving. Venous return is ultimately going to affect cardiac output, as we mentioned, that's Starling's law. So let's talk about blood flow. Blood flow is the volume of blood flowing per unit of time. It's proportional to blood pressure. It's inversely proportional to peripheral resistance. So there's a pressure gradient that exists. It's largest between the base of the aorta and the proximal ends of the peripheral capillaries. So if you think about the blood flowing from the heart, where you have this big, huge elastic opening, very high pressure right next to the ventricles that are slamming out this blood all the time, and then the vessels are getting smaller and smaller and smaller and more spread out, the pressure at the capillary is much smaller. So there's a big difference in those pressures. The cardiovascular control centers can alter those pressures as well. Arterial pressure can rise during ventricular systole. It can peak during something called systolic pressure. It falls during ventricular diastole. It's what we call the diastolic pressure. So you're getting an arterial pressure reading as a systolic and diastolic number, like 120/90. The difference between your systolic and diastolic pressures is called your pulse pressure. It should be around 100. So here again, you see the blood pressure close to your aorta, and then as you get further and further away from your aorta, the blood pressure goes down, and you're no longer able to feel that pulse pressure, the systolic and diastolic pressures from the ventricular contractions. So capillary exchange is the next step on our tour through the circulatory system. This involves the factors of filtration, diffusion, and osmosis. It involves a pressure called capillary hydrostatic pressure, or CHP. CHP is the blood pressure within the capillary beds. The driving force for CHP is filtration. Capillary hydrostatic pressure is pushing water and solutes out of the bloodstream and into the interstitial fluid. So, capillary hydrostatic pressure, pushing out of the blood. Pushing stuff out of the blood. That's what it does. Diffusion is the net movement of ions or molecules from an area of high concentration to an area of low concentration. This is typically faster when there's shorter distances involved, when there's greater concentration gradients, and when the ions and molecules that have to be moved are very, very small. So there's different routes across the capillary walls. Different cells and different solutes are going to take different routes. So the diffusion between adjacent capillary or epithelial cells through pores of special capillaries, like water, ions, small organic molecules, could just go through the pores. Diffusion through channels of the cell membrane would be things like charged ions, sodium, potassium, chloride, etc. Things that go directly through the endothelial plasma membrane would be lipids and other lipid-soluble materials like oxygen and carbon dioxide, which are non-polar molecules. They can just go right on through. Large water-soluble compounds can only enter or leave the capillaries that are found in certain areas of the body, like the hypothalamus, the kidneys, endocrine organs, the intestinal tract. They're only going to let certain things through, and other parts of the body are not going to let these large water-soluble protein compounds. Like think about like carbohydrates or amino acids, those types of things. Plasma proteins normally cannot get through the endothelial lining at all. There's an exception. Those are the sinusoids. Sinusoids are in the liver, where plasma proteins are made. So they have to have a way of getting into the bloodstream. Only makes sense. There's also the factor of filtration. Filtration is the movement of water and solutes out of the capillary. So this is predominant on the arterial end of the capillary bed. When the pressure falls from distance from the arterial until about 2/3 along the capillary, then no net movement in or out will occur. Then reabsorption is the next step. Reabsorption is the movement of water back into the capillaries. This predominates near the venous end of the capillary bed due to a lower blood pressure and a higher osmotic concentration. So more water is filtered than reabsorbed. The difference is returned to the venous system, and it's drained by the lymphatics back into the venous system and can be removed from the body by the kidneys. So again, our arterial end divides into the capillary bed. In our capillary bed, we have filtration dominating on the arterial side, 24 liters a day of filtration occurring there. This is everything that's leaving the blood going to the peripheral tissues. And then reabsorption dominates on the venial side, 20 liters a day. There's a difference, about 4 liters a day that's sucked up by your lymphatics. We'll talk about that in the next, I think it's the next chapter. You can pause the video here, go back and answer these 3 questions. So let's talk about tissue perfusion, the blood flow to the tissues. It's regulated by autoregulation, central regulation. Those are the 2 main ways that tissue perfusion is regulated. So autoregulation would just be changes in the blood flow pattern through the capillary beds. This could be in response to some chemical changes of the interstitial fluid by opening and closing those precapillary sphincters, or by the dilation of blood vessels, the special vasodilators that may be released. Central regulation involves the cardioacceleratory centers, which stimulate the heart, and vasomotor centers, which may regulate the peripheral vasoconstriction using vasoconstrictors like norepinephrine to ultimately decrease blood flow to a particular area of the body. Endocrine mechanisms affect as well, but they have more of a long-term increase in blood pressure and blood volume. But those neural and endocrine are central regulation mechanisms. So autoregulation is just a small kind of localized way of regulating, and then central regulation would be through endocrine and neural receptors regulating tissue perfusion. Ultimately, always getting back to homeostasis and balance. Let's talk about some of our reflexes. We have baroreceptor reflexes. Baroreceptors are those pressure receptors that are found in the walls of our carotid arteries and our aortic sinuses, as well as the right atrium. The baroreceptor reflexes are triggered in response to changes in blood pressure. These involve things like when the vasomotor centers, the cardioacceleratory and cardioinhibitory centers. Responses to these centers basically restore our normal blood pressure. So the baroreceptors will be fired. You see here, baroreceptors are stimulated. baroreceptors are stimulated when our blood pressure rises. And then one of those three things, or all of those three things, are stimulated: cardioinhibitory, cardiocirculatory, or vasomotor center, ultimately decreasing cardiac output, dilating our arteries, and getting our blood pressure to fall. Or vice versa. When blood pressure falls, then our baroreceptors are inhibited. So our vasomotor center is going to be activated, our cardiocirculatory center is stimulated, and cardioinhibitory is inhibited, ultimately increasing cardiac output, and vasoconstriction occurs, getting blood pressure back up, getting us back to homeostasis. You can pause the video here and go back and answer these three questions. So there are several endocrine responses to low blood pressure in our body. You might think, well, I don't know anyone with low blood pressure. Everyone I know has high blood pressure. Well, that's because of our poor diet and lack of exercise. Most Americans have high blood pressure, but as a body, a natural body survival mechanism, our body does not want to have low blood pressure, because if our body has low blood pressure, then our organs aren't getting the amount of nutrients and oxygen that they need. Then our metabolism is not going to be fast enough to sustain our life. It's super important that our blood pressure be maintained at a certain level. Shouldn't exceed that level. Unfortunately, it does for many Americans. So let's talk about the short-term response to low blood pressure. First, the adrenal medullae are going to release epinephrine and norepinephrine. That's going to stimulate your cardiac output, peripheral vasoconstriction, get those blood vessels narrowed, increase the amount of blood, increasing blood pressure. Long-term regulation is done through endocrine mechanisms like ADH, angiotensin II, angiotensin converting enzyme, or ACE, erythropoietin, aldosterone. They're all going to increase blood volume and therefore increase blood pressure. So let's take a look at how these things work. You have a fall in blood pressure. Something happened. A quick short-term fix would be the release of epinephrine and norepinephrine to increase output, constrict those blood vessels, which is going to increase blood pressure and get you back to normal. It might not work though. So a better long-term response would be to send in some endocrine hormones to the kidneys. The endocrine response to the kidneys is going to be for them to release something called renin. Renin is going to be converted into something called angiotensin I in the liver, and angiotensin I goes to the lungs and is then converted into angiotensin II by angiotensin converting enzyme, or ACE. And then angiotensin II is then, oh, here it is, angiotensin II then has widespread vasoconstriction. It causes the release of antidiuretic hormone. It causes the secretion of aldosterone, and it's going to stimulate your thirst. All of those ingredients add to the volume of our blood. The kidneys are also going to, along with renin, they're also going to secrete EPO. EPO tells your bone marrow to make more blood cells. More blood cells going to increase your blood volume. So all of these things increasing our blood volume, ultimately increasing blood pressure to get us back to homeostasis. So increased blood volume and pressure is also going to stretch the heart walls. So in response, our cardiac muscle in the right atrium is going to release something called atrial natriuretic peptide, or ANP. ANP reduces blood volume and reduces blood pressure. It decreases the blood volume and the pressure, the stress of the pressure on the heart wall. So in response, ANP will be, ANP decreases that pressure. And once the pressure goes down, ANP production stops. Sorry, I got a little tongue-tied there. So you have normal blood pressure, then all of a sudden you have an increase in blood pressure. So your heart releases ANP, causing you to lose sodium in your urine, causing you to lose water in your urine, reducing thirst by inhibiting ADH, aldosterone, epinephrine, and norepinephrine, and promoting peripheral vasodilation, all those resulting in a decreased blood volume, decreased blood pressure. So you're going to have an overall systemic decrease in blood pressure and decrease in blood volume. You can pause the video here, go back and answer these three questions. Let's talk about changes in cardiac output. At rest, your cardiac output is somewhere around 5.8 liters per minute. With light exercise, it increases, almost doubling to 9.5 liters per minute. Vasodilation reduces the peripheral resistance, increasing the capillary flow. Venous return increases with more skeletal muscle contraction, and the respiratory pump. The respiratory pump is that increase in volume in our thoracic cavity, which is going to squeeze some of the blood back into the heart. So inhaling, it's going to pull blood into the vena cava. Cardiac output is also going to increase, and blood distribution can be altered even with light exercise. So here you see some of the changes in blood flow. At rest, 750, 250, 1100, etc. But look, when we add light exercise, we're changing. Notice our brain never changes. It's still 750. But look at something like our kidneys. Our kidneys were 1100 milliliters per minute at rest, but now that we're exercising, it's only 350. Our heart is 350 at light exercise and only 250 at rest. Our skin is 500 at rest, but 1500 during light exercise. Why would that be? Temperature regulation, right? Your blood's going to the surface so that it can cool down, and evaporative cooling can cool it off. So let's talk about cardiac output with heavy exercise. Peripheral blood distribution changes big time. Maximum output, 17.5 liters per minute. Holy smokes, that's a lot, right? Considering our initial was 5.8 liters per minute. So you have increased skeletal muscles and skin for heat loss, restriction to non-essential organs like our abdominal viscera. But again, there's no change in blood flow to the brain. Athletic training can improve your cardiovascular performance. It can improve how effective your body does this. Your stroke volume can increase, but your heart rate can decrease. So again, 750 to the brain doesn't change. Heart increases, kidneys decrease some more, abdominal viscera decrease, skin increases, other tissue stays the same, skeletal muscles increase, all with heavy exercise. You can pause the video here, go back and review this section. Let's talk about some arrhythmias. There's something called a PAC, or premature atrial contraction. This can be seen in a healthy person, show no symptoms at all. With increased stress and caffeine, some drugs, it can increase the permeability of the SA of your pacemaker, and therefore make them a little bit more excitatory and cause these premature atrial contractions. Sometimes they're felt as little mini chest pains. There's also PAT, paroxysmal atrial tachycardia. This is like a flurry of atrial activity. Sometimes it's triggered by the premature atrial contraction. The ventricles keep pace, and therefore you have a very rapid heartbeat, up to 180 beats per minute. Ooh, that's a lot. Then you have atrial fibrillation, or AFib, AF. This is when the pulse is moving perhaps around 500 beats per minute, and it produces this kind of quivering atrial wall. The ventricular rate may be the same, so the condition might go unnoticed unless you had an EKG. The PVCs, or premature ventricular contractions, can be common and not dangerous if there's just a few of them, but with increased frequency, epinephrine, caffeine, ionic changes, they can depolarize the cell membranes. The effects can add up. You also have VTAC, or ventricular tachycardia, when 4 or more PVCs without intervening heartbeats occur. This is a more serious cardiac problem. And then ventricular fibrillation, which is responsible for cardiac arrest. This is very fatal, as the ventricles are quivering and they're not actually pumping blood anywhere. This is just showing you some cardiac arrhythmias, your normal EKG, P-wave QRST, premature atrial contraction. You can see there's like an extra little blip right there, extra little blip on the P-wave. It happens a little too close to the T-wave. Here, paroxysmal tachycardia, again, very rapid depolarizations. AFib, that atria is fluttering. Premature ventricular contraction is when you have this T-wave that isn't occurring where it should be along the EKG cycle. Ventricular tachycardia, notice you just have a little P-wave and then lots of ventricular depolarizations, and then VFib is your heart rate's all over the place because it's just fluttering, and notice there's no actual depolarizations happening. It's just a little bit because it's fluttering. I think that's it. Yep, here's the last review questions for the chapter. Let me know what questions you have about the chapter. I'd be happy to go over them with you. And have a great day. See ya.